This video will discuss about biological carcinogenesis. Here mutation is caused by either virus, parasites or bacteria. Virus are predominant. So first discuss about viral carcinogenesis. Based on their nucleic acid content, oncogenic virus are two types: DNA oncogenic virus, RNA oncogenic virus. There are two types of virus. DNA is the genetic material, means DNA oncogenic virus. RNA is the genetic material, means RNA oncogenic virus. So let's start first DNA virus. Mode of DNA viral oncogenesis. This is the virus contain DNA as their genome. This one is the human host cell. Virus enter cytoplasm of the host cell. After that it will enter nucleus of that host cell. DNA present, both viruses also and host cell also. Here two DNA, virus DNA as well as host DNA, both DNA presented. Now two DNAs in nucleus. Host having that blue color one, virus having this red color one. Two type of DNA is present. Now there are the two possibilities. First one is virus DNA replication by using material from host cells. Virus using that endoplasmic reticulum, ribosomes, all the enzyme protein, it causes replication. So here viruses using all the inside materials. Host DNA not replicating. Here host DNA not replicating, holded one. So what will happen next? After replication, the cell burst, host cell burst, host cell die, and multiple viral progeny will come out. This phenomenon is known as replication. Death will occur. So here replication by using material from host cell. Viruses using that host cell material like endoplasmic reticulum, ribosome, all the enzyme and protein, it causes replication. Here host DNA not replicated. After replication, the cell burst, host cell burst, then multiple viral progeny will come out. This phenomenon is known as replication and death will occur. The second scenario, viral DNA integrating. Here viral DNA integrating, host DNA, that blue color and red color one, viral DNA integrating. So this phenomenon is known as integrations. Both of them are replicating here. So here both of them, here virus only replicated, here host DNA and viral DNA both are replicated. So once formed integration, the integrated DNA replicate now. So this phenomenon is known as integration. Because of this integration, mutation takes place. It will causes cancer. So here totally two phenomenon: replication leading to cell death, integration leading to mutation and cancer. Example of DNA virus. There are only 4 DNA virus which are causing cancer. So these viruses are called oncogenic or carcinogenic virus. RNA virus 2 will causes cancer. DNA virus 4, RNA virus 2, totally 6 viruses causing cancer. So what are the 4 DNA virus? Hepatitis B virus, then human herpes virus 8, human papilloma virus, and Epstein-Barr virus. These are the 4 viruses causing cancer. What are the 2 RNA virus? Human T-cell leukemia virus 1 and hepatitis C virus. Then hepatitis B virus and C virus, both are causing liver cancer. So totally 6 are causing cancer in human. Now mechanism. RNA virus entering host. What will happen? See this diagram. This is RNA virus. Here genetic material RNA. This virus enter to human host cell. First enter cytoplasm. After that it will enter to nucleus. After that it will enter to cytoplasm. After that it will enter to nucleus. In nucleus, 2 things are present. Blue color one is called, this one is the DNA, because human having DNA as the genetic material, DNA of the host cell, and red color one RNA of that virus. RNA cannot integrated DNA. Before integrated, RNA is converted into DNA by using enzyme reverse transcriptase enzyme. By using enzyme name is called reverse transcriptase enzyme. Will form a complementary strand DNA. Complementary DNA strand formed. Then RNA of that virus. The complementary DNA will bind with integrated with the DNA of the host. So integration similar to DNA virus. Integration leads to mutation, then mutation leads to cancer. Virus enter to first cytoplasm, then nucleus. Here 2 type of, here DNA also presented, then virus RNA is presented. First with the help of reverse transcriptase enzyme, it will form complement strand. Then both are binding, then mutation will take place. After that it leads to cancer. Example of RNA virus, we already know human T-cell leukemia virus 1 and hepatitis C virus. So complementary DNA resulting double strand viral DNA. This one is called provirus. This provirus integrated into the host cell genome. After that only it will producing transformed host cell. Integration of the provirus bring about replication of viral component. Then it will releasing budding. Now 6 DNA virus and the name of the tumor. First one is human papilloma virus, HPV, human papilloma virus. It will causes cervical, vulvar, penile cancer, squamous cell carcinoma. Then Epstein-Barr virus. This one will cause nasopharyngeal carcinoma, Burkitt lymphoma, Hodgkin's lymphoma. Then hepatitis B virus. It will causes hepatocellular carcinoma. Then human herpes viruses, it causes Kaposi sarcoma, primary effusion lymphoma. These are the DNA virus. These are the things, name of the tumor. Next, RNA virus and it causing name of the tumor HVC, hepatocellular carcinoma. Then HTLV1, it will causes adult T-cell leukemia. RNA virus, hepatitis C virus causing hepatocellular carcinoma. Then human T cell leukemia virus 1 causing adult T cell leukemia. First we will discuss about human papilloma virus in detail. It have 70 genetically different type of human papilloma virus has been identified. Lower risk human papilloma virus 1, 2, 4 and 7 causes benign squamous papilloma in human warts. Then high risk human papilloma virus 16 and 18. They causes squamous cell carcinoma, anogenital region, head and neck. So lowest risk, lower risk human papilloma virus 1, 2, 4, 7. High risk human papilloma virus 16 and 18. These cancer are sexual transmitted disease caused by human papilloma virus. Transmission of human papilloma virus. Now mechanism how it will causes cancer. Human papilloma virus have two protein inside it. It have two protein HBV-E6, HBV-E7. Human papilloma virus having two type of protein. This protein have two mutated gene, TERT and B53. This one having HBV-E7 having two mutated gene B21 RB retinoplasma E2F, totally 4 gene. So when virus enter to human host, after entering nucleus of host cell, the viral protein E6 and E7 mutated these 4 gene. What are the gene? TERT, B53, B21, RB retinoblastoma, E2F. So this TERT inhibit telomerase expression. B53 inhibit B53. This one will inhibit this B21 will inhibit inhibition of B21. Then this one will inhibit RB retinoblastoma E2F. So this TERT, this one will stimulating after mutation the gene will be stimulated. Remaining these three things will inhibiting. After mutation gene inhibited. So these are the three tumor suppressor gene. This one is the stimulator gene. That is the mechanism of human papilloma virus. Expression of two protein HBV-E6 and E7. Each of them two mutated gene. These are the two mutated gene. Two protein mutated 4 gene. This one increased telomerase expression. These 3 gene inhibited gene. E6 gene product inhibit B53 suppressor gene. E7 product inhibit RB suppressor gene. This one is the diagrammatic form. So this one is the host cell. This is the nucleus. Nucleus having DNA. The DNA code for 4 gene. The 2 protein. Okay, 2 protein. It have 4 gene. It have 4 gene. TERT gene, B53, B21, RB. 4 gene presented in the DNA of the host cell. Okay, presented DNA of the host cell. When that virus will enter, this DNA of that virus enter to the cytoplasm first. After that it will reach to nucleus. Okay, after that it will reach to nucleus. After entry the nucleus, it expression of 2 gene. Okay, it will expression of 2 gene. So this virus only expression of the 2 gene E6 and E7. Then E6 will mutated TERT and B53. That E7 will mutated B21 and RB. All gene mutated only things TERT will stimulate. This one will stimulate, remaining 3 things will inhibit. So this is the diagrammatic form of human papilloma virus. Next virus, Epstein-Barr virus. It will causes 6 type of tumor: Burkitt lymphoma, Hodgkin lymphoma, nasopharyngeal carcinoma, some gastric carcinoma, T-cell lymphoma, natural killer NK cell lymphoma. So these are the 6 type of tumor: Burkitt lymphoma, Hodgkin lymphoma, nasopharyngeal carcinoma, gastric carcinoma, T-cell lymphoma, natural killer cell lymphoma. Among them, only this nasopharyngeal carcinoma is derived from T cell. Remaining 5 derived from B cell, B lymphocytes. This one only T cell. Remaining 5 cells from B lymphocytes. Now we will discuss that mechanism. Burkitt lymphoma mechanism of action. Suppose this is mouth of patient and this is a tonsil. Okay, this is the tonsil. Tonsil having cell. These are B lymphocytes in tonsil. B lymphocytes. When Epstein-Barr virus will enter inside that mouth, it have that receptor CD21. Receptor CD21. After binding with that receptor, it will express gene. That express gene name is called LMP1 gene or LMP1 protein. It will go inside host DNA. Okay, that integrated host DNA. Because of this integration, JAK start stimulation started. JAK start stimulation started and B cell will proliferate and proliferate. So their survival increases. They are not die, but this is not leading to any cancer. B cell not die. There are changes due to other reason. It get mutation. Which mutation? So when Epstein-Barr virus enter inside mouth, the B lymphocytes have receptor CD21. Virus coming and binding with the surface of B lymphocytes with the help of CD21. After that, after binding, it will express that protein or gene name is called as LMP1 gene. Then it will go inside the host cell. After that, integrated with the host DNA. After that, it will stimulate. It will stimulate JAK start stimulation started. So JAK start stimulation started and B cell will proliferate. Their survival will increase. They are not die. But this is not leading to cause any cancer. There are chances due to other reason. For reason of other reason, it get mutation. Which mutation? D814 translocation. D8-14 translocation. A portion 14 is transformed to 8. A portion 8 is transformed to 14. So what is T18-14 means? Translocation means 14 is transformed to 8, or a portion of 8 is transformed to 14. Because of this translocation, it can lead to Burkitt lymphoma. In short, Epstein-Barr itself not causing Burkitt lymphoma. Increased chances in B-cell lymphocytes leads to Burkitt lymphoma because it increasing B-cell survival. So this is 8-14 translocation. This is chromosome number 8. This is chromosome number 14. In chromosome 8 have a gene, MYC gene. In chromosome 14 have a gene, immunoglobulin Ig gene. This is the normal scenario. Okay, now some exchange portion. Now chromosome 14 we have two gene. It have two gene, immunoglobulin gene and MYC gene. Now fusion gene are formed. So now fusion gene is formed, because of which increase expression of that MYC protein. So it will proliferate and leads to Burkitt's lymphoma. So summary, Burkitt lymphoma, it is seen in Epstein-Barr virus, not directly oncogenic, but by acting as a polyclonal B-cell mitogen, it set the stage for accusation of 8-14 translocation that ultimately produced full-blown cancer. Next, parasite list and their example. There are 4 parasites which causes carcinogenic in human. First one is clonosis sinuses. It causes liver, bile duct, cholangiocarcinoma and pancreas. Then second one is ophthalmos viverni. This one will causes bile duct. Next one is Schistosoma hematobium. This one causes urinary bladder. Squamous cell carcinoma. Last one is fasciola hepatica. This one will causes cholangiocarcinoma. So these are the 4 parasite will causes cancer. What are the 4? Clonosis sinuses, ophthalmos viverni, then Schistosoma hematobium, then fasciola hepatica. Last one is bacteria. Only one bacteria till date known which causes cancer, H. pylori. That means Helicobacter pylori. It is the first bacterium classified as the carcinogen. It will causes 2 types of cancer, gastric adenocarcinoma, gastric lymphoma. So both of them caused by H. pylori. Both are stomach malignancy caused by H. pylori. The lymphoma caused by H. pylori in the stomach. It is B-cell lymphoma. The name of lymphoma, MALT lymphoma. What is MALT? Marginal zone associated lymphoma. The tumor of this marginal zone of lymphoma, lymphoid follicles. The tumor presented in marginal zone of lymphoid follicles. What is the translocation? Name of the mutation, T1118 translocation. 1118 consequence. Thanks for watching.